Chromosomes start to condense during the interphase stage of mitosis. Here I am showing how the string, which represents the DNA, wraps itself around the battery, which represents the histone, which is a group of proteins. Notice how the string is loose in the beginning, but becomes conveniently tied up around the histone. This is because the genetic information must be organized neatly for later use, not entangled in a mess. And because the duplicated chromosomes are just too long for it to be dangling everywhere, the chromosomes, therefore, must be tightly coiled, forming chromatids. The chromatids are then condensed into sister chromatids, which are basically halves of an X-shaped chromosome. Sister chromatids are identical, and the halves are held together at the centromere. At the end of each chromatid, there is a telomere, which is a DNA structure at the very tip of the chromatid that prevents the loss of genes. Homologous chromosomes are gene pairs, one from mom and one from dad, and they carry the alleles from both parents. These alleles line up on the chromosome in the same order as each other. As you can see, I am drawing a human karyotype of 23 homologous chromosomes. In this case, blue represents genes from the father and green represents genes from the mother. Notice how the chromatids are getting smaller and smaller with each step. Now I am going to duplicate it. When duplicated, there will be two sets of homologous chromosomes. This is what the chart looks like after duplication. Meiosis starts off as a diploid cell, or a cell that has a copy of each chromosome. We end up with haploid cells, where the homologous chromosomes are divided in meiosis 1. Now, when the chromosomes start positioning themselves in prophase stage of meiosis 1, they can sometimes bump into each other. Then, some of the genetic data is traded off. This is where genetic variation occurs. Not only that, but after the chromosome is separated into 8 sister chromatids, the haploid cells are formed in the telophase stage of meiosis 2. The offspring's eye color, for instance, is certainly dependent on the dominant trait, but also the amount of genetic information that was traded off. Now, each gamete is different, in this case sperm cells. That means there are millions of different ways that the offspring can turn out.